Hi, I'm Lori Peterson, editor of Whole Foods Magazine. Welcome to our webinar about a new groundbreaking probiotic study on metabolic endotoxemia, also known as clinical leaky gut. Here to present the dramatic findings is Kiran Krishnan, a research microbiologist and microbiome expert who has been involved in the dietary supplement and nutrition market for the past 16 years. Kiran is the Chief Scientific Officer for Just Thrive Probiotic, and he has a lot of material to cover with you in the following hour. On behalf of Whole Foods Magazine, I'd like to thank Just Thrive Probiotics for helping us bring this information to you. And now here's Kiran Krishnan.
Hi, thank you so much for having me, and thank you all for joining us today. This is an incredibly important topic that I love discussing because I think what we'll come to understand is this is really the core of where many of the chronic diseases start. So we laughingly in a webinar I just did recently, they called it the root cause of root causes. And hopefully that information will be conveyed through. There's some parts of the PowerPoint that are going to look kind of heavy with information, but just know that I'll make sure to explain what is the most important message on that slide. So don't worry about getting lost in the detail as well. But the main point here is I want everyone to really understand the mechanisms by which this metabolic endotoxemia occurs and what are the clinical manifestations of metabolic endotoxemia. And then I'll, of course, also discuss our recently published clinical study showing the impact of the spores on metabolic endotoxemia. So let's jump right in. And when we were looking at developing a probiotic a little over 10 years ago, our whole focus was, what kind of probiotic can we develop for what type of function within the body? And most probiotics on the marketplace today are focused on improving digestive symptoms, things like gas and bloating and regularity. And although those are important things, we wanted to take a different approach and a different look. To us, the most important function of a probiotic is to protect the host. And to protect the host from something that we do every single day that is arguably the most toxic thing that we can do every single day, and that's eating food. So just the process of eating alone causes an amazing amount of toxicity that can then be measured and reflected in the blood and in the circulatory system itself. Now, the types of foods you eat do matter. However, what we see is that when you have dysbiosis and your body doesn't have the type of microbiome to protect you from the process of eating and digesting, even healthy foods can create metabolic endotoxemia. So it becomes extremely important to address the core problem, which is the dysbiosis and the dysfunction within the gut. I'll make sure to give you some information on how a healthy microbiome protects against this phenomenon as well. And then of course, we'll talk about other interventions including the probiotic as well. So first off, what is metabolic endotoxemia? It's a fancy word that basically describes a bunch of toxicity that occurs inside the body after eating food or associated with eating food. To put it more plainly, it's essentially innate immune activation. It's chronic and systemic innate immune activation that becomes subclinical. It becomes extremely persistent. It's at a low-grade level, but it increases and it peaks with more circulating endotoxin. The primary endotoxin we're talking about here is LPS. LPS stands for lipopolysaccharide. Metabolic endotoxemia is a condition that's estimated to affect around 33% of the population. That's if you take the population as a whole, including kids and newborn babies. What we found in our study is in healthy young individuals with no disease of healthy normal weight, the prevalence rate was around 50%. So we estimate, and based on some other information that's available out there, that in populations in their 30s, 40s, and 50s and beyond, especially if you have any sort of chronic disease or symptom, even things like allergies or asthma or diabetes or cardiovascular disease, the prevalence rate is likely much, much higher. The condition is characterized by increased amounts of serum endotoxin, and that's the LPS typically, and that's within the first 5 hours of eating a meal. And the type of meal will increase the endotoxic response. We'll talk about what kind of meals make it worse and what kind of meals could potentially reduce it. Meals that are high in fat and very dense in calories tend to impact the condition more than meals that are low in fat and low in calories. And the type of fat seems to matter as well. I'll elaborate on that in the next slide. And the increase in serum endotoxin concentration is followed by elevations in numerous different cytokines, but the main ones are interleukin-6, interleukin-1 alpha and beta, interferon gamma, triglycerides, and then insulin response as well. Now, it's important to keep these particular biomarkers in mind because these particular biomarkers are the ones that are also elevated in most chronic diseases like cardiovascular disease, type 2 diabetes, and even numerous forms of cancer. Chronic metabolic endotoxemia, and this is something that's happening every day with every meal that you eat, is associated and has been significantly correlated to increase the disease risk of many of the chronic diseases, things like cardiovascular disease, diabetes, even obesity, hypogonadism, that's low levels of testosterone in men, autoimmunity, and even mood disorders like anxiety and depression. And most recently, on September 4th, we now know that Alzheimer's is driven by this phenomenon, by this LPS more specifically. So what is this endotoxin in the first place? Well, again, the full name of it is lipopolysaccharide. The abbreviation is LPS. It is an inflammatory immunogen. That basically means it has the capability to stimulate an immune response, but it also can stimulate an inflammatory response. It is actually a component of gram-negative bacteria in their outer cell wall. So every bacteria in the world can be divided into two groups, either gram-positive or gram-negative. Gram-negative bacteria have this LPS in their cell wall structure. Now, the bacteria uses this for things like adhesion to the host cells so that it can bind and live within the gut. It also uses it as a communication tool, as a way of stimulating the immune system. The thing is, that's important to note, is that the vast majority of bacteria that live in your gut, all of the good, friendly guys, are gram-negative bacteria. So somewhere around 70 to 80% of the good bacteria that live in your gut are gram-negative and contain this highly potent toxin. Now, what's important to note as well is that this toxin, while it's sitting there inside the bacteria, is not really harmful to us. It's not really doing anything that is of great importance. Basically, when the bacteria breaks up or lyses or dies, and this and this LPS is released and it's out by itself, that's when we start to have a significant problem. That's when it acts as a really potent toxin. So it tends to circulate at fairly low levels in healthy individuals, and that's because it's not unusual to continue to have some small level of die-off of bacteria within your gut. So it's releasing some LPS, and some of that LPS will get through into the system, but at a very basal low level, in fact, it's typically very hard to detect, is the normal situation. Now, what happens is when you have metabolic endotoxemia or leaky gut, you basically have a huge increase of this inside the circulatory system after eating a meal. I'll show you how we measure that and what that increase is. Now, the toxicity of this endotoxin is derived mostly by the lipid A portion. That's the bottom portion of it, made up of the fat itself. And bacteria produce this toxin themselves. They manufacture it, if you will, as they're multiplying and dividing. Now, the types of dietary fats seem to matter because that lipid A portion, that portion that I showed you that actually conveys the toxicity, becomes more potent when bacteria use saturated fats to build their LPS. So this is one such study that showed the impact of different types of fats on the toxicity of LPS. One of the things that had the highest amount of toxicity was coconut oil. So you'd be surprised because coconut oil, of course, is known to have health benefits and is kind of the darling of the health industry. But if you have a dysbiotic gut and you're having this metabolic endotoxic response to food and this LPS is leaking in to your system every time you eat, if the bacteria in your gut are making their LPS using fats from coconut oil, they will end up making a more toxic version of the LPS. Now, the toxicity tends to be reduced if they use fats from things like cod liver oil or fish oil, like omega-3 fatty acids. The toxicity of the LPS is actually less. Or even olive oil, for example, tends to be somewhat neutral with the omega-6 fatty acids. But this is the reason why. Not to say that coconut oil is bad. We don't want to necessarily categorize things as good or bad. What we're trying to say is that the fat that's within coconut oil, if the bacteria is making their LPS with that fat, and you have the leaky gut in your body, your gut's not able to protect you from the process of digestion, that LPS is going to get in your system and act in a more toxic way than LPS made with fish oil, for example. So that's what we covered here. And then the very important slide, what I want to describe in this slide is what the LPS actually does once it gets into your body, the type of immune activation that it conducts. And this slide is going to seem heavy, but I'll tell you what the most important points are when we're done with it. First, it's important to know that LPS starts with a ubiquitous pattern recognition receptor, something called toll-like receptor 4. Right? Toll-like receptor 4 is something that's found as part of the innate immune system. It's found in a bunch of our initial first responders, if you will, of the immune system, the macrophages and the dendritic cells. And these macrophages and dendritic cells are all over the body. You know, they're in your circulatory system, they're in your neurons, there's versions of them in your brain. They're everywhere in your body, and they act as first responders. But they contain this toll-like receptor signaling protein, and this LPS seems to function by activating the toll-like receptor system. Now, these cells also have something called CD14, which is another kind of receptor, which binds the toll-like receptor. And between the two of them, once they're bound together, they facilitate the recognition of bacterial proteins or bacterial, what we call epitopes. One of those is LPS. Now, LPS, once it's circulating in the body, gets bound to something called LBP, which is LPS binding protein. And then that complex of LPS and LBP goes and interacts with the CD14 and toll-like receptors, and then that activates that immune cell. So all of this steps basically shows that LPS gets into the system, it interacts with very specific receptors on our innate immune cells, and that interaction activates the innate immune system and produces NF-kappa B. Now, once NF-kappa B is produced, it leads to the expression of a whole bunch of pro-inflammatory cytokines, things like TNF-alpha, interleukin-1 beta, interleukin-6, MCP-1, and so on. So now what we have is the interaction of LPS with toll-like receptor 14, toll-like receptor 4, CD14 on macrophage and dendritic cells, and then that leads to a whole cascade of immune expressions that releases a bunch of inflammatory mediators like interleukin-1, interleukin-1 beta, interleukin-6, and so on. Now, what's important about this slide, of course, the details of CD14, toll-like receptor 4, and all that are not important to remember. What I want you to get out of this is that there's a very specific type of immune activation that is occurring because of the presence of LPS in the system. Now, this immune cascade and immune system has actually, immune response has been very, very well studied, but in the perspective of sepsis. Now, most of us are familiar with sepsis or bacteremia. That's a blood infection. Somebody can go from walking around just fine to being on death's doorstep within 24 hours if they have sepsis or bacteremia. This immune activation is exactly the same thing that happens in the case of sepsis. And so this has been very well studied for that reason. What's important to note here, when this happens with LPS that's coming in from eating food, it's essentially the same thing as sepsis, but it's at a lower level and it's at a more chronic level. Sepsis is a very acute thing that happens. Now, this is at a chronic level and it's at a lower grade level that occurs all of the time. So basically, you can think of it, this metabolic endotoxemia is like a little mini blood infection every time you eat food. That's the most important thing to take away from this entire slide. And also note that it opens up this cascade of immune activation that's marked by all of these key immunoglobulins, cytokines, and so on. Now, the other thing to note is that these same elevations in cytokines are present in many of the chronic diseases I already mentioned, like cardiovascular disease, type 2 diabetes, and so on. Now, what this slide basically shows is in mice that don't have the CD14 capability, they've knocked it out, this whole immune cascade with LPS doesn't occur. So this just reconfirms that LPS works through that same CD14 toll-like receptor pathway that we talked about in the previous slide, and that's the same pathway that occurs in sepsis and bacteremia. So here's how the microbial immune metabolic axis can protect and actually continues to protect the host against this metabolic response. If you look on the left-hand side, it basically gives you some example of positive microbiome metabolites that play a role in protecting the host from this phenomenon. So things like butyrate, acetate, indole, taurine, intactin, these things are metabolites produced by the microbiome that protect the host from this metabolic endotoxemia. There's other things like intestinal epithelial cell derived antimicrobials, things like alpha defensin, lysosome, C-type lectin, all of these things that are stimulated by the microbiome to be produced by the gut lining also play a role in protecting the host from this metabolic endotoxemia. So when you have a healthy, diverse microbiome, when you have healthy communication between the microbiome and the intestinal epithelial cells, there are all of these mechanisms in play to protect us from this phenomenon. So evolutionarily, our body has developed the capability to protect us from this really toxic metabolic endotoxemia. On the right-hand side, what you start to see is perturbations in the microbiome can change the types of metabolites the microbiome is producing, which will then change the response to food in the system. For example, a perturbation could be a single course of antibiotics. That single course of antibiotics throws off the flora and increases the number of mucolytic type of bacteria within the gut. Those bacteria produce more salicylic acid, succinate, and mucinase that breaks down the barrier of the mucus layer. It causes disruptions in the production of all of those healthy metabolites, and that leads to LPS being able to leak in from the lumen into the intestinal barrier, where it causes inflammation. Then the barrier opens up, then all of that LPS goes in. So the point of this slide is just to show that the microbiome really is designed to protect us from this process. Any sort of perturbation in our microbiome can lead to a disruption of that protection. So what are the clinical manifestations of LPS-induced chronic immune activation? We know that this LPS gets into the system. We know that it can activate the immune system within the body. What are the studies that have shown that this LPS actually causes disease within the body? So over the next few slides, I'm going to try to show you some evidence of that. This part of the lecture could have gone on for 50 slides on its own because there's so much data, but I didn't want to cause any sort of death by PowerPoint. So I want to just show you guys a sampling of the studies that have discussed and analyzed this so that you have an understanding that this has been actually well studied. So the first one is virtually any sort of any aspect of metabolic syndrome is better explained by this metabolic endotoxic response than any other phenomenon. For example, heart disease, lipid problems, hypertension, type 2 diabetes, cancers, dementia, the cause of them are all better explained by metabolic endotoxemia than any other theory that's ever been put forth. Now, we also know that people who are obese, for example, have a much higher increased risk of things like dementia, heart disease, hypertension, cancer, type 2 diabetes. What we never really knew is why. You know, we all innately know that if you're overweight, and especially by a significant amount, that your disease risk for your risk for all of these diseases is quite high, but no one could really explain the mechanism by which somebody who's obese actually has an increased risk of dementia or cancer, for example. Now, metabolic endotoxemia explains this quite dramatically, and that's what the next slide shows. So the top part of the next slide basically shows taking people who are of normal weight and slightly overweight and doing high-fat feeding for a period of, I think it was 8 weeks. Then they measured their metabolic endotoxic response to food, how much of this toxin ends up getting into their system after a challenge meal. What they basically showed is that with 8 weeks of high-fat feeding, that the metabolic endotoxic response dramatically increased in individuals. So basically what they're showing is that if you eat excessive amounts of fat and you don't have a good protective microbiome, you're basically going to end up having a more and more potent toxic response to the process of eating food. The bottom part of the slide shows the comparison between obese individuals and non-obese individuals in the metabolic endotoxic response. Basically, what we're showing is that in obese individuals, the toxic response to food is much higher than in non-obese individuals. So once you, in obese individuals, you get this higher toxic response, and that higher toxic response helps explain the increased risk factor for heart disease, dementia, cancer, type 2 diabetes, and so on among obese individuals. So this really affords the explanation, the metabolic and biochemical explanation of why obesity drives increased risk of those diseases, because obese individuals have a much more significant metabolic endotoxic response to food than someone who's not obese. Now, this is something that was published by the American Diabetic Association. What they actually showed that even more than obese individuals, type 2 diabetics have an even higher metabolic endotoxic response. That's in chart A, where they compare the metabolic endotoxic response of obese people. IGTs, people with glucose intolerance, so they are pre-diabetic, and then of course type 2 normal is the dark blue, and then the green is type 2 diabetic. What you see is the green spikes up quite dramatically even compared to the other three categories, because type 2 diabetics seem to have an even more toxic response to food than even just individuals who are obese without type 2 diabetes. So what that shows is this really gives a significant explanation as to why somebody who's normal weight and type 2 diabetic would be more susceptible to heart disease, dementia, and cancer than somebody who's overweight and without type 2 diabetes. So now, again, more and more evidence showing that there's a underlying mechanism here in both of these conditions that are driving these disease risks. And in this case, in type 2 diabetics, the metabolic endotoxemia is even higher. This is another slide that kind of shows the same. They looked at endotoxemia compared to glucose intolerant people, obese people, and type 2 diabetic people. The white part of the bars is the increase over the normal, or increase over obese, or increase over type 2 diabetic in terms of the endotoxemia. So basically this is showing type 2 diabetics and obese people tend to have a much higher response to endotoxic stimuli from food itself. And this slide basically shows that high-fat feeding disrupted the microbiome. So you'll see in that very first bar in figure B, bifidobacteria levels tend to decrease quite a bit with continuous high-fat feeding. And bifidobacteria are some of the important bacteria that reduce inflammation within the gut, increase the utilization of short-chain fatty acids, and so on. So this may be a way of why high-fat feeding in people with unhealthy microbiomes can basically perpetuate the unhealthiness of the microbiome and increase the toxic response. Then the question becomes is, we know that type 2 diabetics and people who are obese tend to have higher metabolic endotoxic responses than people who are of normal weight and non-diabetic, and that's what's driving their increased risk for chronic disease. The question is, which came first? Do they have the higher endotoxic response because they're obese or they're diabetic, or are they obese and diabetic because they tend to have a very high endotoxic response? And there's a few studies that have actually clarified this, and I'm just going to show you one, again, published by the American Diabetic Association. They showed that chronic experimental metabolic endotoxemia, that means causing this metabolic endotoxic response in normal, healthy animals, basically induces obesity and diabetes. So now we know with a high degree of certainty that the endotoxic response comes first. and then the high endotoxic response causes obesity and diabetes. And then once you have obesity and diabetes, the endotoxic response gets even worse, and now you have much higher increased risk for all of these other conditions, of course, including cardiovascular disease and cancer and dementia and so on. Now, what's interesting here is we're showing what is the impact on fat cells when you have this LPS entering the body. And it's really quite interesting because what they show here is when they co-culture LPS with fat cells, adipocytes, what you start to see is a huge swelling of the adipocytes. So somehow this toxin that's derived from the gut that's now entering into the circulatory system because the microbiome and the gut lining is not healthy enough to prevent that migration, we're getting this LPS interacting with your fat cells and actually swelling the fat cells. So it actually causes morphological changes in the fat cells themselves, and we start to see an expansion of the fat cells, and that explains part of why this LPS can drive obesity and an increase in fat mass. This is just another study published in 2012 that shows that the primary insult that led to diabetes is this endotoxic response, this circulating endotoxin that comes from the gut now circulating in the serum in the circulatory system is the major driver and causes the primary insult in those that have type 2 diabetes. So that's just an example of a study that concluded that. Here's another study that showed that LPS, again, this endotoxin induced low-grade inflammation increases insulin resistance irrespective of body weight, because now there's been some issues where physicians aren't able to explain why normal weight people are coming into the clinic that are seemingly healthy. Obviously, they're not obese, so they don't have what we would classically think of metabolic syndrome, but they're coming in with type 2 diabetes in their 30s, 40s, and 50s. And the question is why? We always associated type 2 diabetes with obesity. Now we're seeing that LPS can induce a type of inflammation that can cause type 2 diabetes irrespective of body weight. So you could be totally thin and lean and have a very leaky gut, a very unhealthy microbiome, and every time you eat food, LPS is entering your body and causing immune activation that can lead to type 2 diabetes. This is another study that is a great review paper. This was published fairly recently in 2016, shows the impact of the gut microbiota on inflammation, obesity, and metabolic disease in general. They basically are showing that infiltration of LPS into the circulatory system from the gut after a meal is really the big driver of metabolic disease and all of the associated risk factors with it. This is another paper that another review paper also published very recently, I believe in 2017, shows the molecular link between obesity and cardiovascular disease. You know, as I mentioned before, we always knew that people who were obese had much increased risk of cardiovascular disease. We never really understood the mechanism behind it, because it used to be, it was presumed actually that it was lipid theory. You know, that somehow people who are obese tend to have more lipids in their system, but we now know that that's not true. And the LPS is really the molecular link showing how these risk factors come about and where the risk for chronic disease actually comes about. Here's a couple more slides that just kind of shows you studies. I'll just go through these quick. This study shows that metabolic endotoxemia actually initiates obesity and insulin resistance. It directly increases adipocyte precursors, so it's directly impacting fat storage and fat cells. LPS causes intestinal tight junctions to not function properly, causing permeability in intestinal tight junctions. So this LPS that's generated in the lumen of your intestines is migrating through and actually able to get into the intestinal lining and causes opening up of the tight junctions themselves. So that's an interesting phenomenon explanation for where this leakiness comes from. Elevated endotoxin levels is one of the best explanations now in non-alcoholic fatty liver disease. This is becoming a tremendous problem. I'm actually working with Cleveland Clinic on conceptualizing a couple of studies on this, because what doctors are seeing more often now is this phenomenon of non-alcoholic fatty liver disease. Basically, it's the same kind of cirrhosis that you get in your liver, that used to only be associated with people who were alcoholics, who drank alcohol excessively. Now they're seeing seemingly healthy people of normal weight coming into the clinic with complete dysfunctions in the liver, and when they do scans, when they do their CTs, they see total fat deposition on the liver, and there's no explanation for it. Now endotoxemia and the presence of LPS and all of the immune activation is the best explanation for why this little epidemic is starting. Here's a few more studies that shows that LPS promotes myocarditis. That's an autoimmune type of condition. What they're showing is that LPS in the circulatory system has a way of stimulating an autoimmune response. This is the same kind of thing where induction of autoimmunity in mice can be done through LPS. So we're showing that this presence of LPS plays a role in immune dysfunctions as well, in particular in autoimmunity. Immune modulation of the murine cytomegalovirus induced myocarditis. We know that cytomegalovirus CMV is a chronic infective virus. It's also associated with triggering myocarditis, which is a type of autoimmune disease. What we're showing now is that when you add LPS, this phenomenon, the risk for the myocarditis and the autoimmune disease is increased quite dramatically. This one also shows that if you take LPS and you inject it directly into animals that have relapsed from autoimmune, in this case it was encephalomyelitis, that we were able to get them for the disease to come back just by injecting LPS into these animals themselves. So we know that the presence of LPS has some sort of significant impact on the onset of these autoimmune conditions. Here's a chart. I wanted to summarize a whole bunch of other things for you, but I wanted to do it in a chart rather than show you each one of these studies individually. So I'll just go through this fairly quickly. The first one, leptin resistance. We know that leptin resistance is a major factor in metabolic disease and in metabolic syndrome. We now know that LPS from the gut can actually enter into the enteric nervous system and totally disrupt the communication between the gut and the brain. And that communication between the gut and the brain is what controls leptin function, controls ghrelin function, controls adiponectin function. So that communication gets disrupted because we now know that the enteric nervous system is the conduit in which the gut communicates with the brain through the vagus nerve. Chronic constipation, for example, again, getting into the enteric nervous system, this LPS can totally disrupt how that part of the nervous system works. And we know that the enteric nervous system controls the peristaltic movement and the migrating motor complex in the intestinal, in the intestines. So we now know that this leakiness of the gut, the LPS getting in itself, can be the main driver of constipation and chronic constipation in people. Mood and appetite disorders, we know that there's ghrelin dysfunction as well, similar to leptin depression. We know that LPS can migrate and get past the blood-brain barrier, get into deeper parts of the brain, and actually cause inflammation in the brain, which inhibits dopamine function. It can also inhibit the recycling of serotonin. Those lead to cognitive decline, depression, loss of memory, and recall. We know that it can get into the amygdala, the hippocampus, and can really disrupt cognitive function. You know, and in part, that's part of that kind of the food coma that people can go into, where you eat a big meal and then you're really kind of having severe brain fog and you know, cognitive issues, and that's because the LPS is now entering your system and going into your brain, causing all kinds of inflammation in the brain. And the Alzheimer's study that I mentioned was just published. They've finally shown that this LPS from the gut is what gets into the brain of Alzheimer's people and causes immune activation in the brain, which then kills off the tissue and starts the formation of those beta amyloid plaques that you see in Alzheimer's. So LPS from the gut is the driver of Alzheimer's disease. This was also shown in Parkinson's most recently. They were trying to figure out what is it about Parkinson's condition that causes this neurodegeneration, the dying off of the neurons. They found that LPS from the gut is getting in, embedding in itself into the neurons and causing immune activation in the neurons, which is killing off the neuronal cells, and that leads to Parkinson's. Chronic pain, this is a very interesting phenomenon because a lot of people suffer from chronic pain, fibromyalgia, without any real explanation why the pain is there. What we now know is that this LPS can get into the dorsal root and stimulate something called nociceptors, which send out pain signals on a regular basis. We know that it can affect anxiety by reducing the amount of serotonin in the synapses, by blocking serotonin pumps and serotonin secretion. We also know that it's the best explanation for the onset of anorexia nervosa, autoimmune disease, and of course hypogonadism, low testosterone in men. The gelding theory has been published several times, showing that elevation of immune activation within the body, especially caused by LPS in the system, leads to a reduction in testosterone production in men, especially over the age of 40. So all of the low testosterone, low T that you see being marketed by pharma companies all around is due to immune activation within the system and the subsequent reduction in testosterone production because of that. So the point of this slide, again, to give you guys some explanation as to how pervasive LPS is and all of the crazy things it can affect. I mean, we're talking about testosterone function, we're talking about anorexia here, we're talking about chronic constipation, metabolic syndrome, cancer, diabetes, anxiety, all of these things, autoimmune disease, all caused by these toxins that come in from the gut, entering into the circulatory system and getting into all of these parts of the body, causing systemic immune activation. So just keep in mind how pervasive this LPS is and how toxic it is to the body, and what the really true root causes of many of the conditions we suffer from. This is another great study that showed stress can induce endotoxemia, because stress seems to be able to open up the tight junction in some way. There's many different mechanisms. I could do a whole webinar on how stress changes the microbiome and how that change in microbiome then leads to things like leaky gut and endotoxemia. But basically what they concluded was the #1 cause of workplace absence, disability, and mortality worldwide was due to this permeable intestines and bacterial toxin translocation into the system. So this metabolic endotoxemia is the biggest driver of mortality and morbidity worldwide. That's how powerful this phenomenon is, and that's why we call it the ground zero of most health disorders. Now, how does a healthy microbiome protect against this metabolic endotoxemia? What are the features of a healthy microbiome that can actually do that? These are some of the features. It's not all of them, but these are some of the main features. The first one is a healthy microbiome can actually neutralize LPS right in the lumen of the intestines where it's being released. So it's being released by all these bacteria as the bacteria die off and so on, but it can immediately get neutralized, so it doesn't have the opportunity to enter through the mucosal layer and subsequently enter into the body. So the way a healthy microbiome can neutralize LPS is by stimulating the production of secretory IgA against LPS. So secretory IgA that's being produced by the intestinal epithelial cells in response to a healthy microbiome can neutralize LPS right in the lumen. There are things like increasing PAMP. These are pathogen-associated membrane structures that will then stimulate the immune system to continue to neutralize LPS every time it sees it. The microbiome can also manage the amount of LPS being released and then also try to neutralize it more directly as well. Increase in something called mucin 2. So the mucus layer is the key barrier that protects the intestinal epithelial cells, the tight junctions, from having to deal with things like toxins coming in and then having to deal with toxins going through and into the circulatory system. So the production of mucin 2 is extremely important to maintain the integrity of the cell structure, of the intestinal lining, and the production of mucin 2 is 100% dependent on stimulation from a healthy microbiome. A healthy microbiome can also increase tight junction protein expression, so making sure the cells are sealed quite well. It can also increase the regeneration of affected intestinal epithelial cells, because this layer is only one cell thick. So if a cell dies and it is ejected, it literally leaves a big gaping hole in there. The microbiome and all the stimulus from the microbiome stimulates the regeneration and reformation of that lining. Also, it can reduce inflammation if LPS happens to enter through the system and starts interacting with the gut lining by creating things like intactin, indole-3 propionate. These are things that will actually reduce the inflammatory response to LPS. Of course, short-chain fatty acids are one of the most important things to reduce the metabolic endotoxic response because it does so many things. Of course, I'm not going to go through every aspect of the slide, but the most important things is short-chain fatty acids can decrease bacterial translocation because it tightens up the tight junctions within the gut. It can directly reduce inflammation. It can increase the integrity of the tight junction. It increases the repair of damaged cells of the intestinal lining. It can actually decrease the amount of pro-inflammatory innate immune cells that are being recruited to the lining for inflammation purposes. It can also stimulate the production of mucin 2, like I talked about in the last slide. So it does all of these amazing, wonderful things to protect the gut against this metabolic endotoxic response. And remember, short-chain fatty acids, the primary source of them is from a healthy microbiome. Now, knowing all of this, the idea is that probiotics should be able to help with this phenomenon is not a massive leap, but the question is what kind of probiotic can actually do it, and what kind of features does a probiotic need to have in order to be able to protect the body against this metabolic endotoxic response. When we started working with the spore-based probiotics, we made an assumption that the spore-based probiotics can likely help with this phenomenon. And the reason we were able to do that is because spore-based probiotics have been studied in a similar context in animals for a long period of time. So these are some of the studies that have shown that the spore-based probiotics should be able to help with this metabolic endotoxic response. The first one is, here's a study of Bacillus subtilis that shows that it has a significant function in increasing the expression of epithelial tight junction proteins. All of the proteins, all of the 30 or 40 claudin and occludin proteins that are responsible for maintaining the integrity of the intestinal lining are being stimulated by the presence of Bacillus subtilis, a very important Bacillus endospore. This one also showed actual histological changes in the lining of the intestines with the presence of Bacillus subtilis. So things like villi height, so we're regenerating the lining of the intestines, cell area, cell mitosis, and other things showing that the improvements in the actual structure of the intestinal lining were seen when they were able to add the Bacillus endospores. This is another study that showed the Bacillus endospores improved intestinal barrier function directly by neutralizing intestinal toxins that are part of the normal diet. In this case, it was DON. DON is a toxin that's actually typically found on produce or products that are grown in fields, and that DON is a very potent intestinal toxin, and this Bacillus subtilis is able to neutralize that toxin directly. So based on that, we conducted our own study. It was done by University of North Texas, published on August, I think it was 15th in the World Journal of Gastrointestinal Pathophysiology. What we were able to show is that the spores, just taking it for 30 days without any changes, and basically drastically reduced the metabolic endotoxic response associated with eating food, especially a challenge meal, a high caloric, high fat meal. And basically what we saw was that in the treatment group, we saw a 45% reduction in 30 days in the amount of toxin found in the circulatory system after eating food. So 45% reduction in just 30 days. And it's important to note in those 30 days, we did not have any other interventions in place. No lifestyle changes, no diet changes. Many of these subjects were college students, so they're not doing anything good for them in that 30 days. And yet we were able to see a 45% reduction in the amount of LPS getting into the gut and then getting into the circulatory system, from the gut. And then, of course, all of the associated inflammation with it as well. Now, the scary part is in the placebo group, not only did we not see a reduction, we actually saw a 32% increase in the amount of toxin entering the circulatory system from the gut the second time we tested them 30 days later. So the condition is getting worse. Now, the university that did this study for us had previously published that it takes the body almost 2 weeks to recover from this type of insult from a single meal. So when you look at the spread, essentially the spores were able to reduce the metabolic endotoxic response by over 60% compared to what happens in the placebo group. So quite a significant change in how the body responds to food in just 30 days by utilizing these spores. Now, there were other things that we measured as well. We measured triglycerides. We measured a whole bunch of inflammatory cytokines, and this is just the triglyceride data. If you look at the orange bar, that's the placebo treatment group. The blue bar is the spore treatment group. The placebo-treated group had no change in triglycerides at all. In the spore treatment group, we saw a statistically significant change in triglycerides. In many cases, it was around 40 points on triglycerides. It's a significant change in just 30 days. And the reason for this is because triglycerides is a fat that the liver produces to protect itself. When the liver is under a significant amount of stress, it produces more and more of what we call the blood lipid, triglyceride. So we know, and I talked about it in the case of non-alcoholic fatty liver disease, when all of this LPS enters into the system, it's the liver's job to try to clear it. And as the liver is trying to clear it, it's dealing with all of the toxicity itself. And so the liver is undergoing a significant amount of stress. The fact that we showed a significant decrease in triglycerides completely supports the idea that these spores are in a large part stopping the translocation of this LPS from the lumen into the circulatory system, which is the biggest driver we now know of chronic disease. Now, we also saw, another example, MCP1. You see in the blue bars, as you start treating with spores, MCP1 starts to reduce quite dramatically, and then in the orange, in the non group, the placebo group, we saw an increase in MCP1, which means that more and more immune activation is happening within the gut. So to clarify, MCP1 is a chemokine that indicates that immune cells are being recruited to the gut lining every time you eat food. So what we're seeing is less immune cells being recruited to the gut lining once you start taking the spores. Some of the other things we saw that I don't have the slides for here is reduce insulin response to the food, reduce ghrelin response, so that communication between the gut and the brain. In that chart, remember I showed you leptin and ghrelin tend to get disrupted by metabolic endotoxemia, and that communication between the gut and the brain is disrupted, and so our body doesn't know how to deal with the food coming in, or doesn't really know how to measure the amount of food coming in, so leptin and ghrelin go out of whack. And basically what we showed is a restoration of the ghrelin function, thus a restoration in the communication between the gut and the brain. This is something called a heat map. It's one of the best ways to look at inflammatory cytokines as a whole picture. On the left-hand column, where you see more predominantly green colors, that's a spore treatment group in the second test, so their post-treatment test. And then on the right-hand side, it's a placebo group in their post-treatment test. Now, a heat map shows you the intensity or the concentration of each of these individual biomarkers. In this case, on the left-hand column, you see all of those inflammatory cytokines. The more red, the more intense the color, that means the more intense the levels and concentrations. So when you take a look at the map in general, when you look at the probiotic treatment group on the left-hand side, you see a significant reduction in the redness, in the inflammation, compared to the right-hand side, the placebo treatment group, where the inflammation is really quite significant. You see the intensity of the redness and the prevalence of the redness colors compared to the placebo, compared to the spore treatment group. So this, at a glance, is enough to give you an indication what a dramatic change the spores are making with just 30 days of supplementation on the inflammatory response to eating food within the body itself. Now, again, this same inflammation, especially the top half of the chart, those same markers, interleukin 12, interleukin 1 beta, interleukin 6, are all the hallmarks of cancer, hallmarks of cardiovascular disease, hallmarks of type 2 diabetes. These are the same inflammatory cytokines and metabolites and biomarkers that are elevated in all of those diseases. we're bringing all of those down with just 30 days of supplementation. So then the question is, obviously you can take the probiotics, and that has a significant impact in reducing this metabolic endotoxemia and all of the diseases that are basically associated with that risk factor. But I also want to give you some practical things of what are some of the other things you can do as well. So other recommendations you can make in addition to taking the spores. The first thing is increasing diversity of the microbiome. You want to consider doing time-restricted feeding, so intermittent fasting. I do the 16 and 8-hour protocol. I do it 7 days a week. I fast 16 hours a day and take in all of my calories in an 8-hour feeding window. And basically, studies show that fasting can increase the diversity within the microbiome. Interestingly enough, we got one of the studies back on our spores from ProDigest in Belgium, and that just came yesterday, and I was looking at it, and we confirmed that with the use of the spores, we're actually completely changing the microbiome and the flora within the colon. One of the things we're seeing is a huge amount of diversity increase within the colon just by taking the spores. We're also seeing increases in the groups of bacteria called Akkermansia, especially Akkermansia muciniphila. These are very, very important microbes. High levels of Akkermansia muciniphila is associated with virtually no presence of things like colitis, Crohn's, inflammatory bowel disease, metabolic syndrome, type 2 diabetes. All of these things are associated with high levels, or the risk of the elimination of them is associated with high levels of Akkermansia. We showed in our study that the spores specifically increase Akkermansia. Methodical fiber increases, so you want to slowly add in new and increasing amounts of fiber on a weekly basis. That can actually start to increase the diversity of the microbiome. You don't want to start taking a whole bunch of fiber right off the bat. You don't want to necessarily add 5, 6, 7 sources of fiber all at once into your diet. You want to start methodically adding fiber week to week, every week adding a new source of fiber into your diet, and then on the next week adding in another source without removing that previous source. So by the 6th, 7th, 8th week, you're basically eating 6, 7, 8 different sources of really important fiber, and that really dramatically increases the microbiome. Of course, increasing macronutrient diversity. Our ancestors used to eat 600 to 800 different types of foods annually. We eat somewhere around 15 to 20. If we can increase the types of foods, we will increase the microbiome diversity. That'll go a long way to protecting us against this leaky gut and metabolic endotoxemia. We also want to increase and change our exposure to microbes, so we need to get dirty more. We don't need to sanitize our households. We don't need to sanitize our hands on a daily basis, on a regular basis. We don't need to use antimicrobial bath soaps and things like that. What we need is more exposure to bacteria, and that actually has a drastically beneficial impact on our response to food. We need to eliminate chlorine-based cleaners in the household. Studies have shown households that use chlorine-based cleaners have kids with higher incidence rates of viral infections and asthma and immune dysfunctions as well. Eliminate antimicrobial products, of course, from your household and personal care products. Get a dog. Dogs seem to increase the health of the microbiome with individuals that live in the household, because dogs are inside-outside animals. They go out, they bring in all of these wonderful microbes from the outside and put it into your house. So studies have shown that households with dogs tend to have kids with lower incidence of immune dysfunctions. And then, you can also protect your body from EMVs. These magnetic fluxes that you get from phones and Wi-Fi and all that seem to increase virulence in pathogenic bacteria and viruses within the body that can cause dysbiosis and then lead to further leakiness of the gut. You also want to increase secretory IgA. One of the ways to do that is some of these supplementations like essential fatty acids like omega fatty acids, glutathione, glycine, glutamine, phosphatidylcholine, vitamin C, and zinc have all been shown to help increase secretory IgA. Colostrum can also help with the immunoresponse within the gut itself. And of course, the bacillus spores that I talked about is one of the most potent inducers of secretory IgA and the protective immune response in the gut. Saccharomyces boulardii can also be helpful in this case. And of course, addressing the adrenals. A lot of times, people who are in adrenal fatigue can reduce their secretory IgA production because they have immunosuppression as well. The other thing is resolving leaky gut. Of course, the best and only clinically proven way to resolve leaky gut in a double-blind human placebo-controlled trial is using the spores. There are some studies that show that certain things like glutamine can help as well, but nothing has been able to show a direct impact on intestinal permeability in a double-blind placebo-controlled fashion like the spore. So this is going to be your best bet in reducing leaky gut. But you can also add things like L-glutamine, of course. You can reduce saturated fats while you're trying to fix the leaky gut. Not to say that you want to avoid saturated fats. There are a lot of health benefits to them as well. But if you have leaky gut and you've got chronic conditions, you want to fix that gut lining first before you eat a lot of saturated fats. And like we showed in our study, all it took was 30 days of the probiotic to really make that significant improvement. Lactoferrin is another interesting supplement that can help as well. It can neutralize LPS in the lumen. Also, you can increase mucin production. These are 4 key amino acids that seem to improve the production of mucin within the gut, because mucin are glycoproteins. They're made up of glucose, sugars, and also protein. And these are the 4 main amino acids that make up mucin. So increasing intake of these amino acids can help the production of mucus, which acts as the barrier to protect the body from LPS getting through. Increasing butyrate is absolutely key. One way of doing that is increasing resistant starches into the diet. And of course, the spores. We have shown that the spores increase butyrate production by almost 50% compared to a healthy, normal microbiome. It's probably much higher compared to a disrupted microbiome. And then increasing autophagy and autophagosomes, that's your body's ability to clean itself and get rid of dead and dying cells, damaged DNA, and so on. That increases with time-restricted feeding or intermittent fasting. And in general, butyrate, short-chain fatty acids, that's the key to a healthy immune system in the gut, healthy lining of the gut, and that protective function from the process of eating. So the conclusion here really is metabolic endotoxemia. I always say if it's not the cause of your chronic disease, it certainly plays a significant role in perpetuating the chronic disease. So there are other, interventions and things that people can do, supplements and nutritional things that you can do to help treat or help actually mitigate some of these conditions. And what we know is that if this tap of inflammation continues to stay open, where this LPS from the gut continues to get into the system, with everything else that you might do, you will be taking 1 step forward and 2 steps back. So we have to stop this flood of LPS getting into the circulatory system and causing this immune reaction. And we were very excited to see that just 30 days of the probiotic seems to do that in a very, very significant way. Now we're doing a 90-day version of the study with more participants to further prove this out, but also we want to see how much better it gets in 90 days and how much worse the placebo control group gets within 90 days itself. So that's the overall message here. I want you to understand how pervasive LPS is everywhere it gets into the body, and all of the conditions that it can be responsible for, including most recently Alzheimer's. And we can stop that LPS from getting into the system by just taking the spores. Here are some other things on this slide that you can do to improve that effect, but the spores seem to do it themselves. I also want to mention that there's been significant success in a number of stores that have been carrying this probiotic, and people keep coming back to the store owners and the people that work within the stores and telling them about the amazing progress they've made on so many of the conditions they're dealing with. And now we know exactly why, because they're stopping that endotoxic response. We're stopping that LPS flood getting into their circulatory system. And when you can stop that, you can make such significant progress. If you look at stores like Lori's, which is a single store in Rochester, New York, their first order in 2015 was just 12 units. 5 months later, they were selling almost 300 units a month from one single store. Crystal Lake Health is another store, same thing. They after about 10 months of launching, they were at 144 units per month. Now I know that they're much higher than that already. Zerbos in Michigan, launched in April 2015, started with just 12 units. 6 months later, they're moving around 84 units per month. There's a bunch of stores in Chicagoland area, really wonderful health food stores that we have here called Fruitful Yield. This is becoming one of the top selling products within Fruitful Yield within these stores. I think they have somewhere around 18 or 20 something stores. So we know that there's unprecedented success within these stores with the product. And a lot of that is because of the efficacy of the product, and people keep coming back and telling them all the wonderful things that are happening. So that's the end of this webinar. I'm thankful for Just Thrive for offering this the great webinar specials as well. And then, you know, here's the bottom line and the way you can contact them to get in touch with the product. But again, we're doing more studies. There's going to be more fascinating information coming out. But the whole key to this is let's stop that LPS from getting into the system, because that is a major driver of chronic disease. So, you know, we'll see if there's time for any questions. If we don't, if there are no questions, then, you know, we'll let the webinar folks take over.
Kieran, thank you so much for that informative presentation. And there is someone who's been patiently waiting to ask a question, so we'll let one in, even though it's over time. From what age can this probiotic be used, and in what dosage? Asking in relation to my 5-year-old son with ASD and ADHD.
Okay, great and very important question. With my kids, for example, from about 2 weeks on, they were getting about a half a capsule or 1 capsule a day. So we routinely in the clinics that we work with start with kids at a very young age, even as young as 1 or 2 weeks old, especially if they were born from C-section, if they've had antibiotics right after birth or during birth, and we know that they have a disruptive flora. A 5-year-old child can take 1 capsule a day and do phenomenally well on it, especially if they're on the spectrum, because we know that typically the microbiome of kids who are on the spectrum show an overgrowth of Clostridia spores within the gut. And one of the good ways of getting rid or reducing the amount of Clostridia spores by using these bacillus endospores. So absolutely, from a month onwards, you can start with a cap a day. And if there's an acute reason, meaning there's a cold or flu, or the child has to take an antibiotic, during those acute sessions, you can actually bump up the dose to 2 a day, taken at the same time for a short period of time.
Okay, I'm going to squeeze in one more, and then that's it, folks. What strains are present in this probiotic?
So it's basically Bacillus subtilis, Bacillus indicus, and Bacillus indicus is a very important part of it because it's a high carotenoid producing strain, meaning it gets in the gut lining and it produces high levels of carotenoid antioxidants, about 12 of them in total. And we now know that antioxidant production in the lining of the gut plays a very important role in reducing the damage that occurs to the lining of the gut. In fact, one of the studies on probiotics and ASD showed that antioxidant production is an important feature of a probiotic that could help with ASD. So Bacillus subtilis, Bacillus indicus, Bacillus coagulans, and then Bacillus clausii as well.
Okay. Kieran, on behalf of Whole Foods Magazine, I'd like to thank Just Thrive and you for helping us bring this information to the world at large. I suggest that if you want to ask more questions, you could contact info@thriveprobiotic.com, the email address at the bottom of the screen. Thank you all for joining us and for sticking with us to the bitter end, and have a great day.